All right, so we'll go ahead and get started. Today, Em and I will be reviewing some recent progress in the genetic studies of substance use disorders, much of which we'll talk about today is based on a recent review paper that we wrote together. All right, so today's talk will largely cover what substance use disorders are and how we define them, the epidemiology and genetic epidemiology of substance use disorders, recent progress in substance use disorders genome-wide association studies, and then some conclusions to propose next steps. And so what are substance use disorders? Substance use disorders are psychiatric disorders that frequently co-occur with many other mental health conditions. These can be assessed in relation to a variety of substance classes, including substances that are often described as illicit substances, such as alcohol and nicotine, as well as illicit substances, such as cannabis, opiates, and cocaine. Problematic substance use can result in a variety of negative consequences, including death as a result of consuming the substance itself, or what we call overdose, as well as indirect consequences, such as being involved in a traffic-related accident or experiencing health-related issues and disease as a result of long-term use. And as we'll talk more about here shortly, substance use disorders are influenced by both genetic and environmental factors. And so on the previous slide, we mentioned some adverse health issues related to substance use. And here's a quick example of what this might look like for one substance category, here being alcohol use. And so these are figures from the Global Status Report on Alcohol and Health released in 2018 that reported an estimated 5.3% of all deaths worldwide are attributable in some fashion to alcohol consumption. The pie chart below breaks down these 3 million deaths into different causes, where we can see that nearly 21% of these deaths are due to alcohol-related injury, and approximately 40% due to digestive disease, cardiovascular disease, and diabetes. And so we can see that alcohol use and misuse is having a large impact on the population worldwide, and that is doing so in a variety of ways. And so moving on from the negative consequences to how we define substance use disorders. And so substance use disorders are defined by the DSM-5 as the presence of at least 2 of 11 criteria within a 12-month period, with disorder severity indexed by the number of criteria endorsed, 2 to 3 for mild, 4 to 5 for moderate, and 6 or more for severe. And broadly speaking, substance use disorder criteria correspond to the presence of different substance-related problems and can be assessed in relation to a variety of substance classes, including alcohol, nicotine, cannabis, opiates, and cocaine. And so as I mentioned a couple slides back, substance use disorders can be influenced by both genetic and environmental factors, and these influences are dynamic in that they interact to either increase or decrease one's risk for developing the use disorder, and these factors can be influential across all stages of substance use disorder development. For example, environmental factors such as peer influences, familial environment, socioeconomic status, stressful life events, and co-occurring mental health or health conditions can all serve as environmental risk factors, while positive peer networks, supportive family environments, and greater socioeconomic advantage can all be protective against development of disorder. Similarly, genetic influences can also contribute to elevated risk, as well as serve as a protective role. And so it's also important to note that substance use disorders are highly polygenic in nature, meaning that no one gene is necessary nor sufficient to result in the development of the disorder by itself. And so how can we study the genetics of substance use disorders? Early efforts focused on twin and family studies that demonstrated heritability estimates that say genetic influences account for approximately 50% of the risk for developing a substance use disorder, and that in addition to the substance-specific influences, there are also heritable factors that contribute to substance use disorders more broadly. In molecular genetic studies, we are studying specific points of variation, or what are called polymorphisms. Advancements in genome-wide association studies have allowed us to reliably go across the genome and look at measurements of these specific points of variation and see if these are related to a trait of interest, in our situation being substance use disorders. A similar story has emerged for what was found for early twin and family studies, where there appear to be substance-specific variants that emerge for molecular genetic studies of substance use disorders, as well as pleiotropic variants that influence susceptibility for many psychiatric traits. All right, so now we'll talk a bit about some progress that has been made for substance use disorders. I'll be talking about alcohol and nicotine, and then Emma will walk you through some other additional studies. And so alcohol dependence and alcohol use disorder is a great example to demonstrate the progress that we have made in the past few years. So this figure is from one of the first well-powered studies of alcohol dependence. And to better orient you to it, this is what is called the Manhattan plot, and we'll see a series of these on the next handful of slides. So here on the x-axis, we have chromosome position across the 22 autosomal chromosomes. And on the y-axis, we have the negative log 10 of the p-value. And then these plotted data points correspond to individual variant associations with the trait that we're interested in studying. So basically the line of thinking here is that smaller p-values are going to correspond to these higher points in the plot. And generally, we're looking for 5 times 10 to the negative 8th for a variant to be deemed genome-wide significant. But really what we're looking for are these towers to emerge, which alcohol dependence does a really nice job of demonstrating for us here on chromosome 4. So as I mentioned, this was one of the first well-powered substance use disorder GWAS for alcohol dependence. And here we see that this lone tower emerges in the ADH1B region on chromosome 4 with a p-value of 9.8 times 10 to the negative 13. So this study had about 11,500 individuals of European ancestry that were diagnosed with alcohol dependence. And so that was the biggest then. Let's take a look at the biggest now. And so this is currently the largest GWAS study of alcohol use, which was a meta-analysis of problematic alcohol use in approximately 435,000 individuals. And so one thing I'll point out here is that on the previous slide, we were seeing the most significant variant with a p-value of 9.8 times 10 to the negative 13. And that was the only tower that was emerging. But now with more samples, we're seeing many more peaks, including signals that were in the alcohol metabolizing gene of chromosome 4 that have increased in magnitude substantially. And we also are starting to see additional associations emerge in genes, such as what we see over here on chromosome 2 in the glucokinase regulator gene, which encodes the regulatory protein involved with glucose metabolism in liver cells. All right, so transitioning to nicotine and tobacco use. Here are initial findings from the largest GWAS and nicotine dependence to date, here being assessed by FT and D scores. This study included 58,000 smokers. And so for nicotine dependence and studies of other smoking phenotypes, such as cigarettes per day or regular smoking, there's a robust association with genetic variants within a cluster of nicotinic acetylcholine receptor genes on chromosome 15. That includes the ChRNA5 gene, which is also more specifically replicated the variant RS1696998, which here had a p-value of 1.6 times 10 to the negative 39. So these are the most pronounced results from nicotine dependence to date, but prior studies have found a similar overall pattern of results. And with that, I'll go ahead and hand things off to Emma to tell us about what's been found for other substance use disorders.
Thanks, Joe. Here I'm going to present a Manhattan plot, and this is showing the results of the largest GWAS of cannabis use disorder to date. This GWAS combined data from the PGC, iPsych, and Decode genetics, and included about 20,000 cases and just over 360,000 controls. And this GWAS of cannabis use disorder identified 2 significant loci. There's a locus on chromosome 7, where the lead risk variant is in an intron of the FOXP2 gene. And there's also a significant locus on chromosome 8. And this locus was previously identified by DeMontis et al. In the iPsych data and replicated in the Decode data. And the lead risk variant here is an EQTL for ChRNA2 in the cerebellum, as well as EPHX2 in the cerebellum and adipose tissue. I also want to point out some other results from this GWAS of cannabis use disorder, and this was just recently accepted in the Lancet Psychiatry. And here we're contrasting the genetic correlations between cannabis use disorder and a number of relevant traits and cannabis initiation and the same traits. As you can see, there's some divergence when we look at the genetic correlations of cannabis use disorder versus cannabis initiation or ever use. I'm pointing out 3 in particular here. For body mass index, we see that cannabis use disorder is significantly positively correlated with body mass index. When we look at age at birth of the first child, we see that cannabis use disorder is significantly correlated with an earlier age at first birth, and it is also significantly negatively correlated with educational attainment. When we look at cannabis initiation, we see that there's actually significant correlations with these 3 traits, but in the opposite direction of effect. And actually, for 12 of the 22 traits that we tested, those marked with an asterisk, we see significantly different genetic correlations when we look at cannabis initiation versus cannabis use disorder. Moving on to opioid use disorder, this is the latest and largest GWAS of opioid use disorder. And this combined data from the MVP, Yale Penn, and SAGE. This is by Hong Zhou et al. And they identified a significant functional coding variant within the mu opioid receptor gene, or OPRM1. I also want to point out some interesting results from an earlier GWAS of opioid dependence, and this is by Renato Polamonti et al. He looked at a PRS for risk tolerance, or a polygenic risk score for risk tolerance, and a polygenic risk score for neuroticism. And if we look at the risk tolerance PRS, Renato saw significant associations with two of the contrasts that he tested. So he saw that the risk tolerance PRS was significantly associated when he compared individuals with opioid dependence to unexposed controls, and when he compared exposed controls to unexposed controls. If we look at the neuroticism PRS, we see that Renato saw significant associations with the opioid-dependent individuals and unexposed controls contrast, and the contrast of opioid-dependent individuals and exposed controls. So this suggests that risk tolerance or externalizing behaviors may be more associated with exposure, while something like neuroticism and negative affect may be more associated with dependence than simply exposure. Moving on to cocaine. Cocaine use disorder has had much smaller sample sizes than for other substance use disorders, and fewer GWAS and little replication to date. But what I'm showing here is one of the largest studies published to date, and this is from Spencer Huggett and Mike Stallings. And here they're actually taking GWAS summary statistics from an earlier study by Galerner et al., but here they're conducting a gene-wise association in both the European Americans and the African American sample. And they identified this novel significant gene NDUFB9 in the African American sample. Spencer has also done some interesting work comparing human genetic studies to mouse models. And here I'm showing a figure from a recent paper of his looking at overlap of differentially expressed genes and gene networks when they compared human cases of cocaine use disorder to a mouse model of cocaine self-administration. And they do see significant genetic overlap, suggesting that some of these animal models might be useful for studying substance use disorders, at least in the case of cocaine dependence. Joe mentioned earlier that twin and family studies have identified some common genetic factors shared amongst substance use disorders more broadly, and we see this mirrored when we look at genome-wide data. So here I'm presenting a figure on the right, and this is from a paper by Zhang et al. I do want to note that here we're looking at substance use traits or substance initiation, such as lifetime cannabis use, whether someone has ever reported being a regular smoker. So these aren't substance use disorders per se, but they are correlated with substance use disorders. And we do see genetic correlations between and among substance use disorders as well as substance use traits, starting at around 0.5 and higher. So we do see these genetic correlations mirrored in the molecular genetic literature. We also see that substance use disorders, and in this case, substance use traits, are correlated with other psychiatric disorders. So in the cannabis use disorder GWAS, we saw a significant genetic correlation with post-traumatic stress disorder. Alcohol use disorder and major depression are significantly correlated. And with opioid use disorder, we see a significant genetic correlation with ADHD. So what's next for the substance use genetics field? In our review paper, we outlined 3 key areas that we see as priorities for our field for the next few years going forward. The first of these priorities is increasing the diversity of samples that we include in our GWAS. So most of the GWAS that we presented today, and certainly the largest GWAS to date for substance use and substance use disorders, have primarily included individuals of European ancestry. And it's been shown that when discovery GWAS only include individuals of European descent, polygenic risk scores that are derived from those GWAS have limited utility in target samples of other diverse populations. So we really need to increase the diversity of individuals that we're including in our discovery GWAS. The second priority that we outlined is the incorporation of diverse types of multi-omics data, as well as extension to cross-species data. So in terms of multi-omics data, For example, GTEx is a great resource and one that a lot of us use in conducting follow-up analyses for our GWAS. However, we really need substance-specific data, so gene expression data sets after substance exposure or substance use disorders. Finally, the third priority that we outline is the refinement of phenotypes and ascertainment strategies. So as I mentioned earlier, in terms of cannabis use versus cannabis use disorder, we do see some divergence for some of the substances that we've talked about today in terms of use versus use disorder or problematic use. So for cannabis, there's been some evidence of this. There's also been some evidence of this for alcohol. So when we compare alcohol use disorder to drinks per week or some other measure of perhaps more typical alcohol consumption. But we really need to do a better job of determining whether these phenotypes really show these diverse genetic backgrounds, or if there's something about the ascertainment strategy that is causing us to see these interesting and sometimes paradoxical genetic correlations when we look at use versus use disorder. Finally, in our review paper, we tried to address the question, can we translate any of these substance use or substance use disorder GWAS findings to the clinic yet? And the answer is, unfortunately, we're not there yet. Particularly with substance use and substance use disorders, we really don't have the sample sizes or the robust findings and power that we need yet to be able to use something like a polygenic risk score in a clinical setting. So polygenic risk scores at the moment explain such a small amount of variance in target samples that percent variance explained really isn't at a clinically meaningful level yet, but we hope to get there one day. And there may be other strategies such as pharmacogenetics or drug repurposing, repositioning strategies that may help us translate these GWAS findings to the clinic eventually. Thanks for checking out our video, and we would like to mention that if you're interested in working with us more to better understand the genetics of substance use disorders, please see our PGC website. There's information there, and you can contact any of the co-PIs of the Substance Use Disorders Working Group. We would love to work with you. Thanks.